So maybe I'll just do a quick intro. Anne Cohen is from Hole Oceanographic Institution, and I think we're going to hear a bit about the effects of ocean acidification on some organisms today. And I'm sorry I wasn't more prepared to do that. So Anne, if you would like to share your desktop, then we can get going with this.
Thanks, Drew. So there's a icon here saying show my screen. Should I click that?
That's the one.
And it's only going to show my PowerPoint, not all my personal stuff.
I hope so.
All right. Well, if you see anything untoward, just let me know.
All I can see is a very nice PowerPoint. Okay,
great. Well, thanks so much for the invitation to share this work with you today. And I'm sitting here with my colleague, Dan McCorkle, and we are representing a very much a collaborative effort between Hole Oceanographic Institution and NOAA Fisheries Milford Laboratory. Our colleagues there, Lisa Milky and James Woodman, were actually the instigators of this experiment, although the experiments were conducted at HUI. So we're going to talk today about some results of an experiment conducted in the summer of 2012. And the title of my talk, An Experimental Investigation of the Surf Clam Response to Ocean Acidification, is actually showing the results of that particular experiment. We did run a subsequent experiment in 2013. Interestingly, the organisms, the larvae, did not respond to the feeding. And so that's just aside, just to alert you to the fact that experiments don't always work. This study and our ongoing collaboration is funded primarily by NOAA, but also with substantial funds from Hole Oceanographic Institution and from NSF. So I just want to start by reminding those of you out there that there are myriad of marine calcifiers across the global oceans that use calcium ions and carbonate ions to build calcium carbonate skeletons. And this slide just shows a very small number of those, including fish, which make calcium carbonate odorless. So all bony fish out there are making calcium carbonate structures, as well as mollusks and crustacea and echinoderms. And the problem that we're facing for these calcifiers is really the increase in anthropogenic carbon dioxide emissions. And this plot, taken from the Global Carbon Project report in 2010, just shows how CO2 emissions in kilograms of carbon per year have increased substantially since about 1997. In fact, pre-2000 growth rate of carbon emissions was at about 1% per year. Post-2000 emissions increased substantially to 2.5% per year. So that by 2010, we're looking at global carbon emissions from humans at about 9 billion tons per year. So 26% of those anthropogenic CO2 emissions are actually absorbed by the surface ocean. And what is happening is, as CO2 is absorbed by the ocean, it interacts with water or reacts with water molecules to form carbonic acid. That carbonic acid is not stable, so it dissociates very quickly into free protons and bicarbonate ions. And the declining pH that is known as ocean acidification is caused by this increase in hydrogen ion or proton concentration. But a lot of those protons actually combine with carbonate ions that are already present in the ocean to form bicarbonate. Now, you'll remember from my first slide that all of those organisms are using and depend on carbonate ion availability in the oceans to build their calcium carbonate structures. And when protons combine with carbonate ions to form bicarbonate, that's decreasing the carbon ion concentration of seawater. As far as we know, most of the organisms that calcify are actually using carbonate ions to build their skeletons and can't make use of bicarbonate. This slide just shows, depicts ocean acidification in a nutshell. On the left is a PCO2 on the y-axis over time. On the x-axis, this is the business as usual scenario published in the IPCC report in 1995, showing how PCO2, atmospheric CO2, was projected then to increase over time, hitting a concentration, end of century concentration, just under 700 ppm. I think the current projections are more closer to 1,000 ppm by the end of the century. On the left, on the right-hand side is the calculated ocean chemistry response to that increase in atmospheric CO2. On the y-axis is pH. On the 2y is aqueous CO2 concentrations and then carbonate ion concentrations. And on the x-axis again is years from 1850 to the end of the century. And what you can see is that as atmospheric CO2 increases, the concentration of aqueous CO2 increases as well. And simultaneous with that, the pH of the oceans decline, that's shown in blue, and the carbonate ion concentration also declines. So based on this emission scenario, we're looking at a carbonate ion concentration decline from over the course of this century to about half or less than half of what it was in the pre-industrial era. This is a plot, or a series of plots, published by Martin Steinecker et al. In 2009 in a very nice paper in Biogeosciences, where they used global climate models, couple global climate models, to project the average global decline in oceanic pH and aragonite saturation under a range of IPCC emission scenarios. And those emission scenarios are shown with the colored lines. The black line is based on historical emissions, so we're not going to focus on that right now. Just as a reminder, the aragonite saturation state is basically the product of the calcium and carbonate ion concentration in the seawater in which the organism is growing over the apparent solubility product, which is really the same, the product of calcium and carbonate ion concentration at which that mineral, and we're talking about aragonite, will neither dissolve nor precipitate in seawater. So an aragonite saturation state above 1, aragonite should precipitate from seawater, and at an aragonite saturation state below 1, aragonite should dissolve. So what you see in these projections, hello? I received some sort of message on my phone.
Keep going. We can hear you, Anne.
Okay. So as you can see, in these projections from 1850 to the end of the century, all 3 IPCC scenarios projected a strong decline in aragonite saturation state of the global ocean. And the average decline is about a 1 unit aragonite saturation. None of these scenarios project undersaturation for the average global ocean. But we are projecting a strong decline. And that means it's going to become, the carbonate ion concentration of seawater decreases significantly over the course of the century, and less and less carbonate ions available for those marine organisms to make their shells and skeletons. Oops. There we go. here I'm showing again the projected global ocean average aragonite saturation under 3 different IPCC scenarios, emission scenarios, on the left. And on the right, I'm showing actual data, and these data were put together, compiled by Nathan Rebuck when he was a graduate student in John Hare's lab at NOAA. And this is showing the aragonite saturation state of the coastal ocean today along the northeast coast, calculated from pH and DIC measurements. And what you can see, if you look at the key, what you're seeing is that a large swath of the coastal ocean along the northeast coastline the aragonite saturation state is significantly lower than it is in the open ocean. So if we superimpose a 1 unit decline in aragonite saturation onto these present day values, what we're looking at is that much of the northeast U.S. coastline could be at or under aragonite saturation within the next several decades. So as you recall, aragonite saturation is basically the level of, or the concentration of carbon ions and calcium ions at which aragonite will start to dissolve. And these models are projecting the state for the northeast coastline by the end of this century. Sorry, I'm having a bit of trouble forwarding my slides here. Okay, so recognizing that all these marine calcifiers need carbon ions to build their skeletons, that the concentration of carbon ions in seawater in which these organisms live is decreasing over time due to ocean acidification and will decrease significantly by the end of this century. This prompts us to ask questions about how ocean acidification will affect the ability of these organisms to build their shells and to survive and reproduce as acidification progresses over the century. There's a number of different approaches one can take to addressing this question. What we're going to report on today are results from controlled laboratory CO2 manipulation experiments. And the reason these manipulation experiments are so useful is because we can rear the organisms under controlled conditions in which variables that we believe to be important for calcification can be manipulated singly or in concert to address the following questions. How vulnerable are coastal bivalves? In this case, we're looking at Fissulas solidissima to aragonite undersaturation, and we're specifically targeting aragonite undersaturation because the global climate models project aragonite undersaturation for much of the northeast coast by the end of the century. What, if any, life stages are particularly vulnerable to aragonite undersaturation? And then looking across different experiments and across different species, we can ask what species, if any, are most vulnerable to aragonite undersaturation. There's another factor that we've investigated, and that is the influence of food, or the potential for food availability to either exacerbate or modulate the impact of CO2 on growth and calcification of coastal bivalves. The reason that we're so interested in the food question is shown here, also in a couple of plots on the left showing nutrient concentrations, on the right primary productivity. from another of Martin Steineker's papers, this one published in 2010 in Biogeosciences. And what they're showing are global climate model projections of the decline in surface ocean nutrient concentrations and surface ocean primary productivity, specifically for the northeast U.S. coastal region. If you look at the plot on the right, you can see that these models, and this is the CCSM 1.4 model projection, showing a decline of between 50 and 75% in productivity by the end of the century for this region of interest. So global climate models are projecting not only a significant decline in aragonite saturation state and the common ions available for animals to build their skeletons, but they're also projecting a significant decline in the availability of food. And with this, it's within this framework that we conducted our experiment. So to our list of questions, we've added another one shown here at the bottom in yellow. Does food or food limitation modulate or exacerbate the impact of CO2 on the growth and calcification of our solidissima? So once again, the focus of this study is on the surf clam. It's economically, sorry, we have to ask our students to keep their voices down. One of the reasons we're so interested in the surf clam is because of its economic value, of course. In 2010, it had an average net worth of $28.1 million. It has a pretty broad range along the U.S. coastline, the northeast coastline, stretching from the Gulf of St. Lawrence to Cape Hatteras. And this map here on the right is showing the distribution of pre-recruits. So our study is focusing on the early development, so this map is the most relevant for this particular study. But you can see here pretty broad distribution along the U.S. coastline. And let's have a look at that distribution in relation to present-day aragonite saturation state level for this region. And you can see that assuming a 1-unit drop in aragonite saturation state as projected by global climate models, much of the Spitzula solidissima habitat will be undersaturated by the end of this century. Okay. This study focuses on the development, on early development, which is the development of the larvae from fertilization through 6 days post-fertilization. And one of the reasons we're particularly interested in early development is because the larval shell is made of a much more soluble form of calcium carbonate, aragonite, whereas the adult shell is less soluble form of calcium carbonate called calcite. So I've just showed you here what the adult shell looks like on the left, and what the pre-metamorphic or 6-day-old larval shell looks like in a scanning electron microscope image on the right. And just bear in mind that aragonite is about 50% more soluble than calcite. So this makes the larval shell, and development of the larval shell, much more vulnerable to the projected ocean acidification than that of the adult. Adult males and females were collected around Cape Cod and brought back to the Marine Biological Laboratory in Woods Hole, where they were spawned, and the spawning was induced by elevated temperatures. We did not keep track of specific embryos fertilized by specific parents. Rather, all the eggs and sperm that were released from the adults were collected and pooled in a bucket and allowed to fertilize. Less than 4 hours post-fertilization, so before any shell is made, the embryos were introduced into buckets filled with 15 liters of filtered seawater. Now those buckets were pre-treated with CO2 or air CO2 mixes to target the following CO2 levels: ambient, which is 380 ppm, 1200 ppm, and 2200 ppm. We cultured the larvae at a water temperature of 20 degrees Celsius. We also experimented with 2 levels of feeding. We fed the larvae isochrysis, the specific clay tea. In the high food experiment, we fed the larvae 40,000 cells per mil of seawater, and in the low food treatments, 400 cells per milliliter. The initial stocking density in the bucket was 10 larvae per mil. Now on the right-hand side, I'm showing you the data for the carbonate system chemistry calculations. Now we kept track of carbonate chemistry using spectrophotometric pH measurements and small volume alkalinity measurements. And using those 2 carbon parameters, we can calculate the full carbonate system. Alkalinity, pH, and aragonite saturation state shown here for the replicate buckets at each CO2 level. So I may have neglected to mention that for each treatment, each treatment was conducted in triplicate. Importantly, the chemistry is well replicated in the bucket. The pH and aragonite saturation state are also very clearly different in the different treatments. You'll see here that there's an offset in the alkalinity in the high food and low food treatments. And that, we think, is related to the alkalinity of the fluid in which the algal cells are introduced to the bucket. So this is an artifact. This here is an artifact of the feeding, but it is not impacting the aragonite saturation state. So importantly, the aragonite saturation state in the low food and high food buckets are identical. The cultures were grown up for 6 days, and we subsampled at 3 days and then again at 6 days. And the reason we did this is to look at the impact of CO2 and feeding on the growth of 2 different shell stages. The first stage, the progesticonch 1, is formed within the primarily within the first 3 days, and the second stage, the progesticonch 2, was formed primarily between days 3 and 6. And we were interested to know whether CO2 and feeding had a different effect on those 2 stages of shell development. And the reason we were interested in that is because the mechanisms of biomineralization in those 2 stages are quite different. So the PD1 is formed by the shell gland, and the PD2, shown here, is formed by the mantle. They're both aragonetic at this point. So we quantified growth and development in 2 main ways. The one is by keeping track of shell length, and the shell length is measured perpendicular to the hinge at the widest point on the shell. Parallel to the hinge at the widest point on the shell. The second way we kept track of growth and development is by weighing the shell. So here, the individual shells, and these are about 50 to 100 microns in diameter, are laid out on the base of a Petri dish, and then we use a fine brush, wet with a little bit of methanol, to pick the shells individually from the base of the Petri dish. And those are pooled into batches of 50 shells, because we can't actually get a weight on anything less than 50 shells. They're so tiny. And weighed using a meter-to-liter microbalance. We also kept track of mortality and shell mutations. In these experiments, we're seeing most of the mutations occur during formation of the PD2. So here's two examples of where we're seeing these mutations in shell development in the PD2. Okay, so here's the first of our results slide, and what I'm showing is the shell length of the 3-day-old larvae, or day 3 post-fertilization. Note that what we're seeing here is primarily the growth of the PD1, the shell length of the PD1. On the right-hand side, I've plotted shell length versus targeted CO2 concentration and the actual measured aragonite saturation state in the bucket. The circles are lengths of individual larval shells. The shaded circles are the fed group, and the open circles are the less fed group. So this is high food and low food. Oops. The statistics that we used in this analysis was a 3-way nested ANOVA. We used the nested ANOVA because we are seeing a tank effect, meaning that the buckets are not perfect replicas of each other. What we do see at day 3 is no significant food effect, and I guess this is to be expected because the larvae are not feeding actively by day 3. We do see a small but significant effect of CO2 on shell length. Shells at 1200 and 2200 ppm are different or smaller than shells at ambient CO2. They're not much smaller. The average difference in length is 1.06 microns, or 2% of the length at ambient CO2. We're not seeing an interaction between food and CO2. In other words, the increase or decrease in food availability in the buckets is not affecting the impact of CO2 on shell length. I guess this is also to be expected because at this stage in the larval development, the larvae are not feeding. Here I'm showing the average shell length at day 6 in the culture. So this is day 6 post-fertilization. These images are showing the significant increase or growth of the PD2 in the ambient shells, in the ambient CO2 shells at high food concentrations, and significantly less growth of PD2 by shells in ambient CO2 conditions that are receiving less food. So what we're seeing here is what we're measuring here on day 6 is primarily the growth of the PD2. The plot shows shell length of individual larvae, and I might have forgotten to mention, but we're measuring 50 shells in each bucket, shown against the targeted CO2 level down here, and the actual calculated arachidonide saturation state in the bucket. Again, for high food in the shaded, dark shaded triangles, and low food in the lighter shaded triangles. I've depicted the day 3 length in pink here. So what you can see immediately is that in the high food bucket, 40,000 cells per mil, there's a significant increase in shell length between day 3 and day 6. However, in the low food buckets, there's almost no increase in shell length or no growth of the shells between day 3 and day 6. And once again, the difference between the day 6 low food buckets and the day 6 high food buckets is really reflecting the difference in the growth of the PD2. Once again, we did a 3-way nested ANOVA, and again, we nested it because the buckets are not true replicates of each other. What we find at day 6 is that the effect of food on shell growth is significant, is very significant, it's huge. So that in the high food bucket, the shells are on average 8 microns larger than the shells in the low food bucket. We also see a significant age effect, so that the shells at day 6 are significantly bigger than the shells at day 3 on average. Once again, the effects of CO2, so the difference amongst these 3 CO2 treatments is very small, but it's still significant. At day 6, the shells in the ambient CO2 of 380 ppm are on average 3.45 microns larger than the shells at 1200 and 2200 ppm. That's about 3.5% larger than the shells in the CO2 treatment. We're not seeing any interaction, again, between food and CO2, even though the larvae are eating at this point, and you can see it clearly in the high food experiment. The food is not modulating or exacerbating the impact of CO2 on shell size. What's really interesting to me here is that when we compare the effect of CO2 on shell growth at day 3 with the effect of CO2 on shell growth in day 6, we're seeing that the effect of CO2 on growth actually increases with age. So that, in fact, it's almost doubling. So I've noted this here. The effect of CO2 on shell length is double, at day 6, is double what it was at day 3. And that tells us, that the impact of CO2 on the growth of the PD2 is much greater than the impact of CO2 on the growth of the PD1, meaning that the PD1 is actually quite resilient to the impact of CO2. In both cases, again, day 6, day 3, the impact of CO2 is significantly less than the impact of food. So in a high food environment, even at strongly undersaturated conditions, larval shell growth is significantly faster than it is at ambient CO2 in low food conditions. So food basically is trumping CO2 in this instance. So here's a plot showing, or just summarizing our length data. So rather than showing you the data for every single shell that we measured, I'm just showing you averages and standard errors for each of the treatments. So the shaded circle are the shell length versus aragonite saturation state at day 3 in the high food experiment. The open circle, same day 3 but low food experiment. As you can see, there's very small impact of CO2 and no impact of food at day 3. By the time we get to day 6, we're seeing an increase in shell length, both with age and with food. Again, a very small impact of CO2. So in summary, there's no food effect at 3 days, but the effect of food at 6 days, once the larvae are eating, is 4 times greater than the effect of CO2 on shell growth. Now we're looking at shell weight. Once again, we're picking the individual larvae, dried larvae, from the base of the Petri dish, pooling them into batches of 50, and measuring them on a microbalance. This plot is showing shell weight versus the calculated aragonite saturation state in the buckets. I'm only showing you the shell weights on day 6. And what you can see, once again, the effect of food in the dark shaded triangles versus low food in the open triangles. Here we're seeing a significant food effect on shell weight, such that the fed shells are on average 2.3 micrograms heavier than the less fed shells. We're not picking up a CO2 effect on shell weight. And we think one of the reasons is that the effect of CO2 on the development of the shell is so small that we're just not picking it up in the weight, whereas we did pick it up in the length. And once again, we're not seeing an interaction between food and CO2, so that food is neither exacerbating nor modulating the impact of CO2 on the weight of the shell. We also looked at mortality and abnormalities, and the types of abnormalities I showed earlier. These are the data. So here I've plotted the percent of dead shells at day 6 versus the targeted CO2 concentration, and the percent abnormal shells that we saw at day 6 versus the targeted CO2 concentration. And in neither case are we seeing a significant impact of either carbon dioxide or food on either mortality or abnormalities on day 6. There is a small trend in the percent, a slight increase in the percent abnormal shells in the low food bucket, but this trend is not significant, not statistically significant. Okay. Finally, what we've done is collected as much data as we could find to compare the response of our Cicera solidissima larvae to aragonite undersaturation with that of other northeast coast species and also some from the west coast. So here I'm comparing, we're comparing the weight of the shells at undersaturation as a percent of the weight of the shells at ambient CO2. And the data set is probably smaller, definitely smaller than the number of studies out there. And one of the reasons is because very few people are actually collecting weight data, weight or calcification data. So this is really representing what is out there right now from a number of different studies. So the blue are all Chrysostria, different species, Virginica, Ariakensis, and Gigas, all larval shells. This is a C. Virginica adult shells from one of Justin Reese's experiments, and those were published in 2009. These data are agapetin irradiance from one of the experiments that we ran here at Hui. And here I've plotted S. solidissima, this study, low food, and high food experiments. So what we're seeing is that in all cases, undersaturation appears to have a negative impact on the shell weight or the calcification of both larvae and adults of different species. In the case of Spiciulus solidissima, that effect is actually not significant. So, whereas it is in the case of the other species. So we're concluding that S. solidissima is potentially more resilient or resistant to ocean acidification than these other species. At least we believe that it is probably the most resilient of the species that we've worked with today. Okay, so I'm just going to wrap up here with a summary of our observations. We can say that the effect of aragna undersaturation on shell growth is small, but it's significant. So we're seeing an effect on the shell length, and we're not seeing an effect on the shell weight. We see most of that effect in the development of the PD2. So we're concluding that the PD2 part of the early shell formation is more sensitive to aragna undersaturation than the PD1. We see that the effect of food limitation is significantly greater than the effect of CO2 on development and growth of the larvae. But we're also seeing that there's no interaction between food and CO2, so that food doesn't modulate or exacerbate the impact of CO2. We're sticking our necks out and suggesting that Spiciulus solidissima is more resistant than other species studied to date, and that's based on data for which there are weight measurements. So thanks. That's the end of our presentation. And Dan McCorkle is here, and we'll both take your questions. Thank you.